Creating an expression vector for interleukin-6. Interleukin-6 is a pro-inflammatory cytokine that's involved in various physiological processes in the body. For example, the immune response. It's usually produced by macrophages. Elevated levels of interleukin-6 have been associated with tumor growth and metastasis. In this presentation, I'll be talking through the creation of an expression vector for interleukin-6 that can be used to study the role of interleukin-6 in tumor progression. First of all, I'll start off by building the vector. For the vector, I'll use a pcDNA3.1 expression vector as the backbone. This plasmid contains a multi-cloning site with various restriction enzyme sites, which allow for easy insertion of DNA. It also has a CMV promoter for high levels of expression and an ampicillin resistance gene that can help in bacterial selection. I will start off by performing PCR on a template plasmid for interleukin-6. I will design my own primers for this, and the primers will include restriction sites for HIND3 and ECO-R1. These are two enzymes that are found in the multi-cloning site of pcDNA3.1 and will help the PCR fragment slot into my plasmid backbone. After performing PCR, I'll carry out restriction enzyme digests on both the PCR fragment and the restriction enzyme digest in order to remove any debris that may remain within the DNA and also open up the vector. Then I'll carry out gel electrophoresis and perform a gel extraction purification step to further purify the DNA. Next, I'll ligate my PCR fragment into the vector using T4 DNA ligase. Transformation into bacteria. I will transform competent E. coli cells with my vector in the next step. To do this, I'll add some of the ligation reaction to competent E. coli cells that have been incubated on ice. Then I'll heat shock the cells for around 30 seconds before returning them to ice. This will allow the E. coli cells to take up my vector. I will then incubate the E. coli cells in SOC medium at 37 degrees Celsius on a shaker overnight to allow the cells to replicate. Then I will plate the cells onto ampicillin plates and incubate them and allow colonies to grow. Any colonies that grow on my plates will contain my plasmid, as my plasmid contains the ampicillin resistance gene and allowing them to grow on the ampicillin plates. I'll validate my expression vector after this. I'll pick colonies from my plates and amplify and purify them by doing a mini prep. Then I will perform a number of restriction enzyme digests. My backbone, my pcDNA 3.1 backbone contains a Beagle site, and I will utilize this by performing various combinations of digests with either HIN3 and Beagle2, or EcoR1 and Beagle2, etc., and then run the digests on an electrophoresis gel and analyze the band sizes produced. I will then look at the sizes of the bands to see if it's what I would expect for the vector I was trying to produce. Obviously, in this example, my PCR fragment isn't in the vector, but so in my expected vector, the fragment sizes would be a bit different. Another step I can take to verify my plasmid would be to send it for Sanger sequences. This will allow me to detect any minor mutations that may have occurred during the whole process and confirm 100% that the plasmid I've produced is what I intended to produce. Finally, I'll transform my vector into HeLa cells. HeLa cells are an immortalized cervical cancer cell line. I will do this by liposuction. I will use lipofectamine 2000 reagent, and I'll add my purified plasmid DNA to this reagent and incubate it. Then I'll add the mixture to some plated HeLa cells and incubate it for 1 to 3 days. After this time, the cells should have taken up my plasmid DNA, and it will be ready to analyze via methods such as ELISA.